time is here. Welcome to this celebration of this year's iGEM team. iGEM, what is it? It stands for International Genetically Engineered Machine, nothing less. The easy is, it is a competition, international competition in molecular biology and synthetic biology. And there are thousands of students around the world participating in this competition, hundreds of teams, and they are using synthetic biology to try to find solutions on challenges, locally, globally, like challenges in the environmental, the toxicology, the pollution, health problems, medicine problems, food, agriculture, using synthetic biology. And this competition has been going on since 2003. And the University of Oslo has participated in this is in 10 years. That also needs a celebration and a piece of cake after this presentation. The important thing is to say that University of Oslo is, let's say, is the host for the team by the Department of Biosciences. And that is run by Professor Dirk Linke and researchers Athanasios Saragliadis. And I'm also a co-supervisor. But the point is that the team, they define the project, the research questions, what they really want to solve out there. That is not driven by the supervisors. It is not every year that we have the gold medal. I have asked the team if they can tell about the project they had been working with for 1 year. Detectimol, I have asked them, what is it to take to achieve the gold medal in this competition? Because it's not only a clear, like one gold medal, but the team will explain that. And also a little bit about how this work has been, 1 year at UI of Oslo iGEM. We have made this cake for you to just say, well, it's not a piece of cake to participate the iGEM year. But Lynn and the staff and everyone, we have been using the night making this beautiful cake that you will have a piece of afterwards. And we are streaming, so welcome to all of those listening to us by Dag Bakker, he's behind here. So everything has to be on the microphone. So please, team 24, you're welcome to the stage. You can
Okay.
Okay,
Yeah. Wow. It's so good to be here at this point of our journey. And let's just get to it maybe and begin introducing everyone.
Yeah. So, hi, we are Detectimol, or two of the team members. My name is Marlene Metz. I'm a second-year master student in molecular biology and biochemistry. And this is...
I am Mariana Jodevandelou. I'm a third-year master student in toxicology and environmental sciences. And
we're going to talk a bit about our project Detectimol, which is a continuous cortisol monitoring device. We're also talking a bit about what is iGEM and how did we get to this point. So first of all, iGEM stands for International Genetically Engineered Machine. It's an international competition for synthetic biology with, by now, very many participants. It uses the genetically modification of organisms to solve global or local issues. And they have split the competition into different villages, which are thematic fields for the different types of projects. So for example, we are part of the diagnostics village, which is the red heart that you can see here in the back. But there are many different fields ranging from bioremediation to the foundational advanced village. Now, this is the picture from above an iGEM tradition they take every year of all the participants. It's not everything, there's more on this picture. I had to crop it off to fit it on the slide. This is from this year's 2024 Grand Jamboree in Paris. Everyone who participated in person, it's more than 4,500 people there. Back 21 years ago when they started, it's been very small. It's been in Boston, just a handful of people. And yeah, because we're wearing red pullovers, I managed to spot us. That's us. Yeah, but what exactly is synthetic biology? It's a bright field of biology that covers engineering principles and biotechnology techniques like DNA sequencing, genome editing, to modify organisms and then create new biological parts such as sequences of proteins.
Yeah, and it is quite important to tell you why, for people that are interested at least, why you should join and why we joined maybe and what we got out of this. And we have to say that the most important part of iGEM was that we got our own hands-on experience. So we were part of something that hadn't been done before. We got to do laboratory work that was completely our own. We got to work on something exciting and new that hadn't been done before, as I said. And we got to go to conferences. We went to Denmark and talk to other people that work with iGEM. We got to make connections with them. We got to know people from the synthetic biology field and biology field in general. And yeah, the most important thing for me, and I feel for a lot of the members, was also leading our own research project. So this was very exciting because we got to be part of a team, which was challenging in its own way because you get to coordinate everything from start to finish on your own. And that was hard, but it was very giving as well in the end because everything from the experience was ours. And of course, our own publication through the wiki that we wrote. And it's something that has our own name on it. And it's something for the next people to read when they want to know about what we did. So if you're interested, having your own wiki, your own words out there is, I think, the most important thing. It's not a master's or a bachelor, but it's your own work. And that's very exciting. Yeah. See? So for the people that want to join and are interested, there is going to be a team meeting in January of 2025. And this is the course number if you're interested, and definitely try to join. We feel like the main message of this project has been achieved, or we tried to work towards achieving it, and that's making a better world with synthetic biology. Yes, just quickly to show you, this is our team name and the village we were part of, the Little Heart. Detectimol is a name that we went through a lot of different phases to change the name together with the team, but detection molecule is basically the main core of our team name, so that's where it's inspired. And a little bit about the background of the inspiration. So when we looked into different topics that we could cover, one of them was that 140 of every million people suffer from Addison's disease. That's when too little cortisol is produced in the body. And 70 out of millions of people, every million people, have Cushing syndrome, which is when too much cortisol is produced in the body. And what we noticed with this, the common denominator here, the common marker, was cortisol. And even though it's a molecule that affects us in all types of ways in our bodies, it affects us in many different functions, we noticed that it wasn't really as covered as we would like. So we dubbed it as the overlooked molecule, because even though a lot of research had been done, not a lot of monitoring projects, and even though there was, like, as I said, articles, but nothing concrete there. And we wondered, where is their monitoring device? So we compared the different methods here. We went through sweat, saliva, and blood, and we noticed that sweat and saliva, even though they could be easily collected, and there were a lot of articles for sweat and saliva, unfortunately, only sweat had a beta program made basically of a monitoring device, but saliva a little bit more difficult, mostly with articles that we saw. Precision still was blood. So the best precision was offered by blood testing, which is done to this day for people who are suffering from these diseases. However, none of these methods offered continuity, which is what we wanted to do, continuous measurement. And that's where the name Detectimol, a continuous monitoring device for cortisol, came to be.
So next to Addison's disease and Cushing's syndrome, we realized cortisol is also important for something else that we all can relate to, and that is stress. So we conducted a little survey. We managed to reach 60 people from all over the world, mostly from Europe, but also from South America and some from North America, Asia, and even Australia. Unfortunately, we lack complete response from African countries, but we did manage to reach 60 students from all over the world to reply to our survey about stress and how they feel about stress in regards to their studies. We also asked them what type of gender identity they have, just to see how diverse is the background that we're working with. And it's mostly female, but we do have replies from all types of people. So the first question we asked them, what type of degree are you pursuing? Are you still in high school? Are you doing a bachelor's or master's degree or a PhD or something else? And when are you planning to submit your thesis, if you have a thesis? And not surprising, most of the replies were from bachelor and master students who are going to submit their thesis in the next 2 years. Then we asked them, how do you feel in regards to your overall studies? How stressed do you feel on a scale from 1 to 6? With 6 being the most and 1 being barely any stress or not stressed at all. And in general, people are not feeling too stressed about their studies, but when it comes to their thesis submission in particular, the closer the time comes, the more stressed they are, which is also not really surprising, but up to 50% say they feel extremely stressed when the thesis is going to be submitted in the next 1 or 1.5 years. And the ones who have more than that have a lot lower stress levels. So we asked them, what else do you feel stressed about? What types of things stress you out apart from studies? It's also work, health, time management and responsibilities, personal relationships, social norms and interactions, the future, financial stability. It goes through all types of things. So we asked them, would you consider wearing a continuous monitoring device for cortisol to measure your stress, to manage your stress? And 2/3 said yes, they are open to the idea, or they would definitely wear something like this. Well, approximately 1/3 said they're a bit skeptical, or they would not wear it. Now, for the biophysical mechanism, we're getting a bit into the science. This is the glucocorticoid receptor, ligand binding domain, so it's just a part of the protein, and it binds cortisol specifically at this ligand binding site. When the cortisol binds, we have a conformational change, so the 3-dimensional structure of the protein changes, and that shifts the electron density of the protein. And we want to use that. We want to bind it to a gold electrode, and when we have the shift of the electron density, we change the impedance of the electrode, which changes the electrical signal that we want to measure from that. And this we want to put into our design of a cortisol patch. This is our first approach to doing this, and we hope that it looks something like this, which you may know as the glucose monitoring device for diabetes patients. Our design has 4 important parts. First of all, the microfluidic system from where we get the sample. It's 2 tiny needles pinching slightly through the skin to get interstitial fluid, which is the fluid directly under the skin, and it contains cortisol. Then the cortisol is led to the detection protein, which is number 2, directly attached to the gold electrode, which is number 3. And then a computer chip is supposed to read out the signal, number 4. So the plan of this project has 6 steps. First of all, we had to design what we want to do, and then the next step is cloning, expressing, and purifying the protein of interest, so the ligand binding domain of the glucocorticoid receptor. And from there, we can use the protein to demonstrate that it binds the ligand, as we theoretically already know it does. And once we have shown and demonstrated this, we can continue with the proper electrode design, test the microfluidic system together with it, and develop the microchip. So we managed to get to this point, number 3. We were working with the demonstration of the binding when we went to the Jamboree.
Okay, so We didn't stay idle. We tried to show you a little bit of our results here. So the most important thing that we felt like we saw in our laboratory work towards the end of our project was that we managed to get a purified version of our protein of interest, and also have that visualization with a Western blot of the purified protein of interest here. And we're very happy. It's just 2 gels that you see here, but it's a lot of work, a lot of designing, a lot of building blocks that need to go where they need to go in order for this to work. Just for us to show you these 2 pictures, so we're very proud of it, and we hope that you see a little bit of it here. We didn't stay idle after we had this wiki freeze, as we call it, where it's a deadline where we have to deliver a big chunk of the things we found so far, the things we've told you about. When this time comes, we can't do any more edits until now, at the end of the project, where they give us some time to do some edits after the competition is finished. But we really wanted to keep working with the purification of our protein to see if we could improve the yield and the purity of our protein of interest, which we did manage by changing the running buffer recipe, by changing also the protocols for the chromatography from gradient to isocratic, and increasing the protein residence time. And again, like a simple graph perhaps, but we managed to see by what we did in the changes in protocol, we managed to see a good change, a positive change, and something that we can work forward to. And it's not perfect yet, but we feel like it's better than before, and that's where success is. And of course, as Marlene mentioned, we really wanted to continue working with step 3, where this is where microscale thermophoresis assay comes in, MST, which is about making sure that we can prove the binding of the protein of interest with its ligand. And that's kind of a work in progress, but we're very happy. We've been in contact with Björn Dahlhus, who has let us use the machine, and we're keeping in touch for future uses as well.
Yeah, so now we're getting back to the negative impact. This all sounded very nice, and we're very happy to have reached this, but we also had a third of people who said they might not want to wear a patch like this. And we were asking them, what are your concerns? And we also got input from Anamika Chatterjee from the Digital Life Norway. She asked us to reflect and think about this. So we did some thinking, and we managed to come up with the ethical considerations of the data usage. So what happens with the data when you wear such a patch? Who can read it out? Who can use it for what? We wanted to make sure that this data is just for the patient and maybe the healthcare provider to prevent abuse, misuse, and potentially even discrimination based on personal stress levels. And another thing that we have to consider is the risk for health tolerance, which means the body may not react properly to the device because it does pinch slightly through the skin, and this is some testing that has to be done with the final design. Another part of iGEM is, apart from the science, to explain the science, to make it accessible for people. So we took part in education as something we wanted to contribute to, and we were invited by the Norsk Teknisk Museum to present our project at the Forskningstorget, the science fair, in September. In these pictures, you can see us using our educational concept to explain to visitors of all ages, really from primary school to their parents, other researchers, more than 2,000 visitors, what we have been doing and why. To give you a bit of overview, because I think it's really fun, what is our education concept? We wanted to explain what is a receptor protein, what is a ligand, and what is synthetic biology, and we used Lego for this. So we designed a little bit of a protein that you can build with Legos yourself with these instructions we would call the plasmid. And if you use the plasmid to build a protein, it makes you yourself the ribosome. And in the end, you come up with a protein that can do a conformational change to trap a little rubber, and the rubber is supposed to not be a Lego brick to symbolize the ligand, cortisol, which is not an amino acid like proteins are made of. We also wrote a little short story to explain what is cortisol and what is the endocrine system in the human body. We translated it to 5 languages. We have English, Norwegian, Greek, German, and Spanish, just so everybody can, in a playful way, understand what we did and why.
Yes, so we are very happy to also have done something that iGEM calls human practices, but in maybe more simple terms, it's basically the exchange of information and learning from others and listening to researchers and the relevant audience to basically build the project and the idea better. Everything for us started when we contacted actually Professor Martin, who is from a Canadian team, an older iGEM team, who had worked with cortisol detection. So we reached out to him. He was very nice, and he guided us through the beginning steps. And then we went through many, many different researchers, which we've tried to put in a little a linear timeline here, that helped us basically keep developing and keep working with this idea. And then we reached a little bit before the end, we talked with Jens Petter Berg, who was very generous in helping us at the last steps before we went into the laboratory to confirm our idea and to give us that little push to keep working with what we were interested in, which was working with interstitial fluid and the cortisol detection patch. And then towards the end, we didn't just get feedback on laboratory work, but also with Rein Osland and with Nikolai and with Gebrand, who is also here today. We got a lot of feedback on marketing, how to talk with people, how to pitch, and how to basically inform the audience about what we're doing and why it's important and how it's important as well. Yes, so a little bit quickly, we didn't just talk to people one-to-one, we went to bigger audiences and actually used the information and all the feedback we got, both from our professors and as of course Kirsten, who introduced us to the master students and gave us that possibility to kind of go out there and talk to people who are in the beginning of this journey and maybe are interested in joining iGEM. So we worked with just presenting to different master programs such as pharmacy, pharmacy, it was chemistry and biology, yes. And we also collaborated with other teams, other iGEM teams, such as the one from Taipei, called Digiworm. We were very lucky, this is us in a meeting with them, to kind of collaborate and to get ideas of how it is to present and things that we could improve and vice versa. And towards the end, we were part of the opening of the UIO iHub, which is also part of the university, and we were invited by UIO Growth House and INCH, and we were part of a pitching competition with 5 other teams, and we received the public project pitching jury award prize of 20,000 kroners before we went to Paris for our project, basically, which we thought was very nice. It showed faith in the project, and that was, that meant a lot. Okay, so we went to the Jamboree. We went to the 2024 Jamboree in Paris. What happened? And the results were, I don't know if everyone knows, but we won gold. There was a lot of screaming.
Let's do it. Drum roll.
Exactly, we're very happy. I think I think I've shown everyone the gold medal in person because we're very, very proud of it, and we can't believe it actually happened. So, yeah. So just to wrap up, this is a little bit about the Jamboree experience in Paris. So we got to meet the CEO of iGEM, which is very interesting. And we met him in different occasions, and he was just very open and very down to earth. It was very interesting to talk to him and get in contact with him and just talk about the competition. Part of the competition also meant that we had to have a stand and talk with different people, with judges, with press, with just many visitors that came to our stand, the other team members from other teams, and explain what we're working with. So maybe 8 hours a day, some 4 hours a day, it depended. But I mean, I didn't want to go from the stand. I wanted to talk with everyone. So it was very cool. And here is also another picture of our team in Paris. Here is a little bit after our judging session. This is a very important part of the competition where we met the judges and we had a 5-minute presentation to kind of give them an extra idea after we had the wiki freeze, as I said, where we gave the results, what happened after. And we felt like it went very well as well. And then we had another live stage presentation. So there's a lot of things that fall into place here. This is in front of the whole people that everyone from the Jamboree could join. So the whole audience was just people, judges, and other members of other teams where we presented. And then our very proud picture that you can see officially taken with our gold medal. Too many bright smiles here in the picture, I think.
Yeah, and this is the end. We have to thank a lot of people, especially, of course, the Science Library for having us, for having our backs, for letting us present today and celebrate us today. Then we have a bunch of sponsors, actually. Digital Life Norway and IDT helped us out. Of course, the department Ivogene, the Teknisk Museum, and we are very happy to also put ING and UIO iHub on there because we were invited to their pitching project competition. Mm-hmm. The 2 QR codes, if you're curious, one of them, the one for Instagram, is for the UI Oslo team. So it's not just us, it's the previous teams from this university the last 10 years. And everything that's yet to come. So if you're curious about what is coming in 2025, I recommend checking this Instagram channel out. And the red one, is our personal wiki. So if you're curious about our project and want to read more, you're very welcome to do that there. Yeah, and at the end, this is our team. Those are all of our 7 team members and our 3 lovely supervisors. So thank you so much.
Thank you so much.
Thank you. We have time for some questions, but I also want to say, did you say your names?
Yes, in the beginning.
And we have 3 more here at least. That is, you have to raise your hands. Bror, Espen, Sigurd, and Sigve. Mm-hmm. They are here also. So you can see them here on the picture. Yeah, I did ask you. It was you did have gold, so you did go through a lot of stuff. But other teams would also have gold. So that means that my question about what takes it to do that, is that you have to deliver on not only the lab, and that is the most important, the molecular or the synthetic biology, that you have a product, and you do that in a scientific way, but also all of this outreach and having the scientific conversation with scientists out there. So you have to meet a lot of, let's say, tasks to get the gold medal. But then, do you have questions from the audience? There are no students here that want to participate next year. Espen can ask. Why are we so great? That's because you are great.
Very good.
So the point is also, as Marianne said, that you can. Sorry, there is. Yeah, thank you.
Yeah, my name is Jens Petterberg, and I was very happy to be contacted by you and to have some questions regarding the more, let's say, clinical aspects of the project. And so, first of all, congrats. And I think it's also a very important issue because this is something that one has been struggling with for years and years on how to really have a continuous measurement of cortisol, in particular in patients with a loss of cortisol production, that is adrenal insufficiency, because it is very difficult to mimic the diurnal variation of cortisol when you give the synthetic cortisol to these patients. So by using this, I think, and also developing these instruments, so to speak, will be very important for this quite rare but a very important group of patients. And also, as you took up the aspect of stress, and also stress research uses a lot of cortisol measurements and as a proxy for, let's say, how stressed you are in a given situation. So I think that is also a very important aspect of your research. But I was wondering whether you have actually been in contact with patients who have either too little or too much cortisol.
That's a very good question, I feel. And it was something in our project that we heard from our supervisors and from our judges that it was a part that we didn't cover as much. We talked with people that were either in a stress situation or people that took our survey, but we didn't specifically ask people that were suffering from either Addison's or Cushing's. It was part of our goals, and we really wanted to achieve it. So it felt like we were missing that core part there, just to hear about their opinion on the project and what they would think would be interesting if they would be willing to wear the patch. Not only people who are suffering from either chronic stress or everyday stress, like we addressed in our survey, but we feel like it was something that we really wanted to do, and we definitely want to do in the future, because that will be the most giving, I think. Yeah.
I could just add that user involvement in research is a topic that is discussed and also in medicine, and we are not, let's say, good enough to take that aspect into our consideration when we do research. But and it's also discussed at what level and how this could be done in the most efficient way. But anyway, if you then develop this, let's say, product further, I would be happy to assist you in getting in contact with the patient organizations, which I know some representatives of. So
Perfect.
best wishes.
Thank you as always, Jens Petter Berg.
That was very good.
Yes.
Bror.
I just wanted to give a comment and said that we had been posted on the Addison's Association in Canada for people with Addison's, and they were quite optimistic about the project. So that is something, at least. Yeah.
Yeah, because I was thinking about that. If I was a student having an exam, if I wanted to go with a stress monitor, I can understand that some of them did not want to. But in a pathological situation or closely to, it would be, as you say, yeah, very relevant. So, Dirk, is there something to add? I want to add that you will have 10 grants or points if you participate in this. So if you have some questions about iGEM, you can contact us.
Yes.
And then we have a piece of cake. Lynn tells us to come. We have to cut in that beautiful cake with the Detectimol logo. So thank you, and have a nice piece of cake and a cup of coffee.
Thank you so much.
Thank you.